In this video, we will cover the clustering step when analyzing Visium spatial transcriptomics data in Chipster. In this step, we compute clustering in the selected PCA space and use the nonlinear dimensionality reduction method called UMAP to visualize the computed clusters. So in clustering, we are dividing spots into groups based on their gene expression. We apply the same graph-based clustering approach as we did for the single-cell RNA sequencing data. We use the principal components from the previous step instead of genes, and we select the number of PCs for the PCA space, and then we compute clustering in this space. We use the shared nearest neighbors approach to draw edges between the spots and form the graph. We then use the Louvain algorithm to cut the graph into clusters of spots. Now let's take a look at how to use this tool in Chipster. We can look at the elbow plot from the previous step to help select the number of PCs to use for the clustering step. Remember from the single-cell RNA sequencing data analysis tutorial videos that we can look for gaps or for the plateau in this plot to help select the number of PCs. In this example, we choose 30 as the number of PCs, but you can also try out different numbers of PCs. We can select the Seurat object from the previous step and use this clustering tool for this step. We choose the number of PCs here, and we can also choose the resolution parameter value that sets the granularity of the clustering. If we increase the value of the resolution, it will lead to a greater number of clusters, and we should make sure that this value is appropriate for our dataset. We can then run the tool. This tool outputs the Seurat object with the clustering results, as well as visualizations of the results. The first plot shows a UMAP visualization of the data where spots are colored by their clusters. The last plot shows the clusters on top of the tissue image. After clustering, we can also use the Visualize Clusters tool to visualize specific clusters of interest.